Hi folks, today we'll be talking about chapter 19, our third macromolecule, proteins. So up to this point we've talked about carbohydrates and lipids. The last of the three macromolecules that make up the basis of nutrition would be proteins. This goes a little bit into more than just necessarily food-based proteins. We'll be talking about how we use them in our bodies, and eventually these will become enzymes that our bodies use to go do all the work that they do in our bodies. Quick side note here, amino acids are pretty common in terms of the types of questions asked on like NCLEX for nursing students and things like that. So while for us as a online course, it is understood that this is going to be kind of an open notes situation, the learning and memorization of your amino acids is kind of one of those things that a lot of students are expected to do. So I do strongly encourage you to start working on um, learning the side chains of the amino acids for actual memorization. There are a lot of really good kind of like smartphone game resources for this, especially because it is on things like the MCATs. It's very easy to find a lot of study tools for it. So let's get started. If we're going to talk about proteins, we first have to talk about amino acids. And amino acids are going to be the building blocks of proteins. We can kind of think of a protein as like a little Lego building, and the amino acids would be the individual Legos in the building. There are 20 amino acids, and like we said, proteins are made up of amino acids. They're called alpha amino acids because the amino group is in the alpha carbon, which is next to the carboxylate carbon. So here's this basic kind of setup that we're going to see for an amino acid, and then if it's a protein where we have multiple amino acids strung together, this is simply going to be like an amide polymer. So if you go back to chapter 16, where we talk about amides and how they're linked together in a polymerization, that backbone is the backbone of proteins. And then each amino acid contains a different R group within it. There are two different ways, I guess you could name, well, three different ways to name amino acids. One, its full name. There's also a three-letter abbreviation, which is utilized when we're talking about codons, and there's a one-letter abbreviation as well. So we're going to go through the different R groups. So essentially, each amino acid has, like we said, the same backbone to it. The only thing that changes is this R group on the alpha carbon. So we're going to go group by group to talk about the different amino acids and kind of the characterization of them. So we have neutral, nonpolar. These will be R groups that particularly contain like alkyl groups. Neutral but polar, so these would be compounds that contain polar portions to them. So things like hydroxyl groups or sometimes like a thiol group or something like that. We can have basic R groups, so anything typically being like amino based, and then we can have acidic R groups as well, and these will typically be carboxylic acids that we see for that. Okay, learn all the amino acids. Like I said, most secondary degree testing will ask you something about amino acids and do expect you to know the R groups for those. So either making flashcards or a fun Quizlet or something like that will be very helpful here. So we're first going to start with the neutral nonpolar side chains. As you can see, each of the amino acids presented here have the full name, three-letter abbreviation, and the one-letter abbreviation. They're not always the greatest at being equivalent to each other, and part of the problem is that we have um, amino acids that are very similar to each other and similar in name that you can't always give them like a good abbreviation. So we have glycine, which is the simplest of all the amino acids and is the only non-chiral amino acid as well. If you notice here, the alpha carbon here is not chiral because there are two hydrogens attached to it. Pretty much every other situation we deal with, the alpha carbon is chiral. Alanine, valine has a V shape to it. Leucine is just valine with an extra CH2 group. Isoleucine would be having that basically the V in an isometric. So at the third carbon here, kind of an addition. Phenylalanine has a phenyl group. Proline is another weird one because proline is cyclic. If you see here, the R group actually binds in on the amino side of the backbone. Methionine, these are all neutral and nonpolar. From here we can go to neutral, but polar side chains. So as we said, these would be things that contain hydroxyl groups or other types of charged, polarly charged electronegative portions to the R groups. We have our basic polar side chains. So these will be things, again, that would be good bases. So these would be things that would, in a proper solution, act as a base, or we could have acidic side chains as well. Okay. So as we mentioned earlier, 19 of the 20 amino acids have the alpha carbon as chiral. We talked about glycine, this is the only non-chiral one. With very few exceptions, most living systems are going to be the L form of the amino acid. So again, if we're talking about D and L, we're looking at, the chiral carbon furthest away from the carbonyl, which would be this one right here. Here's our alpha carbon. Please keep in mind glycine, because it's achiral, is not going to be D or L. So most amino acids are going to exist as what we call a Zwitter ion, which is a really fun word to say, but basically it's a dipolar ion. So it's kind of like its own acid-base reaction, right? Because when we think about an amino acid in this kind of backbone, right, we have an acid on this side and we have a base on this side. So it's like doing its own acid-base reaction, where here's the acid donating the proton, here's the base accepting it, where we get something that looks like this. This is how we normally see amino acids, not like this. Now, if we have a string of amino acids together building a protein, it will only be the ends, the beginning and the end will have be the Zwitter ion. It's the polymer in between is all neutral. So because we have these Zwitter ions, we have to kind of talk a little bit about pH and what exactly is happening. So we have something called an isoelectric point, which is going to be the pH at which any particular amino acid has a net charge of 0. So depending on what type of group we have, that will determine where, they're happy, essentially. So neutral atoms will be in a net charge of 0 at about 6 pH. Acidic, shouldn't be shocking, is going to be less than 6. Low pH for acidic, higher pH for basic. At any pH value that would be above the isoelectric point, the amino acid would have an overall negative value. So if we had a neutral amino acid at pH of 8, there would be an overall negative charge on that. If we go below it, it would be a positive charge. So if we had a neutral, again, amino acid that was at pH 4, that would have a net positive charge. Because of these Zwitter ions, right, the fact that it's its own acid and base kind of reaction happening, amino acids tend to act as buffers, and they can react with both hydronium, H3O+, or hydroxide. Because of this, most Zwitter ions, right, amino acids are going to have similar properties to them. They're typically white crystalline solids, have relatively low melting points compared to other solids, but are also very soluble in water. So that solubility in water comes from the fact that these are highly polar, right? This is cation anion going on here, which we know water loves to hang out with. So we didn't talk about it too much when we were doing our chapter 13 functional groups, where we touched upon the concept of a thiol. And this is why we touched upon it. So there are amino acids that contain thiol groups, and they're very, very important because these thiol groups can do something called disulfide bonding, where they form what we call a disulfide bridge. And we'll learn in a little bit about how two R groups, two amino acids would interact like this. But for right now, let's just say two amino acids that are perhaps on a very long protein chain in the presence of oxidizers can form disulfide bridges. And these disulfide bridges are a very strong way to kind of connect this side of a protein to another side of a protein. So we talked about this idea that we could have like an amide peptide, right? That we're going to have just kind of this repeating pattern, this repeating backbone, and we're just going to decorate the the alpha carbon in different R groups to represent the different amino acids that are all being linked together. So to do this, we're talking about a peptide bond, which is basically going to be the combination of a carboxylic acid and an amine, which, as we know from chapter 16, is an amide bond. So we have the amide linkage here, which would be the link between the nitrogen and the carbonyl carbon. When we make this link occur, we know that we also produce water as a byproduct. So a dipeptide, as you can guess, just like we had disaccharides, a dipeptide is a compound that is made up of two amino acids that are joined by that peptide linkage. Because if we had two different amino acids, for example, here we have glycine and alanine, we could get two different peptides out of it because the sequence is ordered. It depends on whether we went glycine then alanine or alanine then glycine. Because remember, it's not equal. One end is going to have the NH3, the other end is going to be the COO minus on this side. And so what ends up happening, here's the H, right? So remember, glycine is our non-chiral amino acid, so the R group is just an H. So they tend to draw it like this. I prefer, when I am drawing it out, to draw it with the H pointed down, just like we did with the alanine here for the methyl group. It kind of helps remind me that there's multiple R groups there, so multiple amino acids. So when we name dipeptides, it's simply just naming the two unique acids together. So either glycoalanine or alloglycine. Again, two different compounds made from the same starting materials. So a peptide would be a short-chained amino acid polymer. So, maybe like 20 or so. A polypeptide is going to be intermediate in length, typically under 50. There's some wiggle room there. A protein will be a polymer with more than 50 amino acids. So what we're talking about here is, right, we're knitting that polymer over and over and over again, and we're just adding and decorating with the R groups. This is simply a conventional thing so that everybody is on the same page. We will always write and read our peptides from the N terminus, so the N side, to the C terminus. So we're always going from left to right in this direction. Partially this is done for helping with naming things. So as you saw in the previous slide, whichever one came first, with the name. But it also will become important when we start looking at protein synthesis as well as DNA synthesis, that we have a clear understanding that everyone is looking at it from the same angle. So obviously there are some very, very important little peptides. So these are relatively small. For example, vasopressin is only 9 amino acids long, but vasopressin is super, super important. We produce a lot of it when we're sleeping, and it's so you don't pee while you sleep. And here you can actually see that disulfide bridge, which is also really nice. So that disulfide bridge, there are two cysteines here, and it helps form this little like loop in the polypeptide. But what's neat to see here is here's oxytocin. Oxytocin is very, very similar to vasopressin, not only in shape, they both have this disulfide bridge to give it kind of this loop to it, 9 amino acids long, but they're at the 3 and 8 carbon, we have different amino acids, and so they do very different things. One is often a part of, labor and delivery, and the other one stops you from peeing at night. So it's really incredible when you start learning about proteins and peptides, how just small changes can have vast effects on the purpose of a protein. Peptides, like we said, can get significantly longer. Here we have a hormone, essentially, that is released by the pituitary gland that helps with other adrenaline gland related processes. And again, here's our N terminus. So we're reading from N terminus to C terminus. This is the length of it. So here's just kind of a list of these peptides, relatively small peptides, and what they do. Where they come from and what action they have. So we already talked about oxytocin. We already talked about vasopressin. Other things here would be stuff like prolactin, gastrin, glucagon, HGH, human growth hormone, right? So a lot of these, either through puberty or fetal growth, are a lot of these many, many smaller ones. But we also have things like insulin, which is another small one. So when we're looking at these in terms of their size, right, this starts getting kind of crazy because up until now, we've been talking about relatively small molecules. When we started getting into our macromolecules, right, our lipids, our carbohydrates, our proteins now, we're starting to get larger and larger numbers. So now we have molecular weights that are, in the thousands to hundreds of thousands in size, in mass. So when we're describing something as a protein, what we're saying is we have a very large natural polymer. Some of these, like we said, the molecular weights of some of these could be several million, mass particles in mass. They can get very, very large, which can be overwhelming, and we're not going to be memorizing any of that kind of stuff. Really what we want in terms of memorization, the best for you would be to be very, very comfortable with the amino acid side chains, how the protein backbone looks, and just be comfortable knowing how we build those peptides and how we break them apart. Um, so, because they're so large, these proteins, they're not usually something that gets to go explore other parts of the body. Heart proteins stay in the heart. Liver proteins stay in the liver. They're stuck there. They're too big to pass through cell membranes. So this is often a first pass when triage is occurring for a patient. One of the first things that we can do is check urine. If we find protein in the urine, that's typically a sign of kidney damage. If we find heart enzymes in the blood, this is a sign often of a heart attack. So we can type enzymes and proteins relatively quickly and have some good ideas about triaging patients based off of that. The proteins that we are going to see in our bodies, in animals, where we're, acquiring some proteins and amino acids from, they will all be in the form of zwitterions. You should be comfortable with the concept of an isoelectric effect, as well as, what happens to specific amino acids at different pHs that are either above or below. their isoelectric effect, or excuse me, isoelectric point. So again, proteins have a ton of functions within our body. Really, proteins are the movers and shakers of how business gets done in a body. Everything from catalysis, structure, um, protection, to movement, to transport, to kind of central nervous system stuff. Proteins do it all. So proteins can act catalytically or structurally. So a catalytic protein has its own name called enzymes. We'll actually be talking about enzymes in a little bit. Other types of proteins would be things like structural proteins or storage proteins. We also have protective proteins. We are more familiar calling these antibodies, right? These are the little soldiers that help maintain and protect our body from invading viruses, bacteria, and other, foreign substances. Um, Other proteins include things like regulatory processes. So this would be hormone regulation. What would we uptake? What will we downtake? Depending on what our needs are. Nerve impulse. So one of the biggest things is proteins that are basically myelin. So they're the coating around our central nervous system. Proteins for muscle, obviously, that's one most people think about, and then proteins for transport. So we have proteins that will kind of bind and drag along small molecules to wherever they need to be in the body. Proteins come in two shapes. We have fibrous shapes and globular shapes. Fibrous shapes look very, very similar to like pull and peel Twizzlers. That's the best way I can describe them. It's kind of like woven fiber. Things like collagen, keratin, elastin, they're very structured proteins. So things that make up our hair, make up our nails, things that help stiffen our skin. These are all these fibrous proteins that are very rigid. If you've been paying attention, we talked about the idea that all of these proteins are way too big to, be passed along to another area, go through a cell into another area in our body unless there's damage. So if you've ever bought in a very, very expensive moisturizer before that has collagen that touts that it will help replace collagen lost in the skin to make it firmer and all that kind of stuff, you're just smearing that on your face. It's not being absorbed in. Globular proteins, on the other hand, are typically going to be like these blobs. Um, Literally, that's what they are. Now they have pockets to them, and pockets and crevices where important things happen that we'll talk about, especially when we get in enzymes. But they aren't as structured, and they're not nearly as rigid. We can have simple proteins, or we can have what we call a conjugated protein. A conjugated protein will contain amino acids plus other things. We call these other things prosthetic groups, but sometimes it's a metal, sometimes it's like a whole other group, like an organic chemical molecule. So here is an example of hemoglobin. Hemoglobin is this molecule right here, all the bits and pieces of it, but you can also see the heme group in the hemoglobin. And the heme group is the portion of the hemoglobin that is responsible for transporting the oxygen around. And in that case, the heme would be the prosthetic. There are many, many different kinds of prosthetics. Some of them will be, like we said, metals, some will be phosphate groups, some will be lipids. Basically, anytime we have a protein that is conjugated, there's some process going on that's helping regulate, and oftentimes that prosthetic group is part of how it gets regulated. So we're going to start talking about how we build proteins. We know that amino acids are the building blocks of proteins, but they're much more complicated than just stringing together a bunch of amino acids. Um, the first pass at the structure is what we call primary structure, and that chain of amino acid sequence is actually just all it is. Um, the primary structure will dictate everything else. It will dictate the shape, whether it's fibrous or globular. It will dictate whether the protein, whatever the protein does. But it's very, very important to understand that the primary structure is simply the string of the different amino acids. So here we have an example for you in blue, that backbone of the amino acid, and then each of these R groups would be a different amino acid R group on the alpha carbon that we would add to it. So to give you an idea of how big some of these proteins can get, we have here insulin, which is pretty low in terms of things, 51 residues long. Residue simply means, an amino acid building block. So something like human growth hormone, 191, and then we get into gamma globulin, which is 1320 residues long. So some of them get quite large. Beyond primary structure is secondary structure, and this is basically a lot of intermolecular forces occurring within the string of that initial primary structure. So here it's really going to be kind of a lot of hydrogen bonding that goes on. We know that we've got this repeating backbone pattern where we have carbonyls and amino groups present. And so those will be interested in hydrogen bonding with each other. And when they do that, they can form two possible shapes. They can form an alpha helix or a beta pleated sheet. So here's an example of the alpha helix, and the way it works is on this stream here we can think of as that backbone, that blue backbone that's a repeating pattern, and then the R groups are along for the ride here. But basically the carbonyl carbon oxygen of one of the atoms will hydrogen bond with the amino group of another amino acid further down the line. And as this happens, it kind of torsions the overall primary structure into a secondary structure, in this case of an alpha helix. A beta pleated sheet, the best way I can describe this would be if you took a piece of paper and you kind of folded it into an accordion, like you were gonna, like you were gonna make a fan. That's kind of the general shape that these take. So here's just kind of an example of a random coil of material, just so that we could see what the beta pleated sheet looks like versus what the coil looks like. So here's our alpha helices, here's our beta pleated sheet. In both cases, the intermolecular interactions are happening at the backbone. That is the primary takeaway lesson from Unit 3 for secondary structure, is that the secondary structure occurs when looking at the backbone. Tertiary structure, on the other hand, really results from the interactions between the different R groups, or those defining qualities that make glycine glycine and methionine methionine. Different types of these interactions that we can see would be things like disulfide bridges, which we already talked about. A salt bridge, this would be where basically an acidic amino acid would form an ionic bond with a basic amino acid. We could have hydrogen bonding occur, if they're two polar molecules, or we could have hydrophobic exclusion interactions between nonpolar residues. So here's just an example of the different kinds of tertiary structure, or those R residue interactions that can occur. Disulfide bridges, salt bridge, hydrophobic interactions, hydrogen bonding. These can all happen, and they all do. So again, think about where these proteins exist. Oftentimes they're in our bodies, which is going to be a very water heavy environment. And we know that some of these amino acids that make up the protein are hydrophobic, right? They're non-polar. And so what oftentimes ends up happening as the protein folds up on itself and kind of takes on the final shape that it's going to take, oftentimes the protein amino acids that are hydrophobic will kind of interact with the hydrophilic ones in a way that um, force the hydrophilic ones to be on the outside of the folded protein so that they can interact with the water solution that they are in. And the hydrophobic ones are pushed more towards the center to be excluded. So we've talked about primary, secondary, and tertiary structure, and all proteins have that. All proteins have a primary, a secondary, and a tertiary structure. However, not every protein will have what we call a quaternary structure. And a quaternary structure is just kind of like repeating patterns of a subunit to form a bigger protein. So in the case of hemoglobin, there are four different monomers of the subunit that all come together to make hemoglobin. And so that's why there's quaternary structure of hemoglobin, but not every compound will have a quaternary structure. So here again is our hemoglobin. We've color-coded the different units to make it a little easier to see all four of them, but they're all identical to each other, and it's just those four atoms together, or excuse me, those four proteins together make up one hemoglobin. And so you can even see in the renderings of these backbones for the amino acids that we have multiple double helices. This looks like a beta pleated sheet over here. So you can see tertiary structure in a quaternary structure. So obviously we do ingest proteins. It's not just something we make, we ingest them as well. And so there needs to be a process by which we can break apart the complex protein and turn it into something smaller that we can begin to digest and utilize bits and pieces of. So in a lab, we would do this under acidic conditions with water. In our bodies, this is also done by whatever digestive enzymes are, assigned to whatever specific proteins we're looking at. But basically, it still is the same general process, just done much more efficiently with enzymes through digestion. But we have some sort of protein, it is broken down into smaller proteins, smaller peptides, and then from there it's broken down even further into the base units, the amino acids, the individual Lego blocks, so that they could say, okay, go use these amino acids for something else. The last topic we're going to talk about with proteins would be denaturation, or kind of breaking a protein. Part of denaturation is also when a protein can no longer do its base function. So a denaturation of a hemoglobin would be a hemoglobin that can no longer carry oxygen around the body. So when we have protein denaturation, what we're doing is breaking apart that, primary, secondary, and tertiary structure. And when we do this, what ends up happening is biological activity stops functioning. If this is what the tertiary structure of this protein was supposed to look like for whatever function it had, as it begins to get denatured, we start interrupting those tertiary and secondary interactions, and what we're left with instead is just kind of a garbled mess of unwoven proteins. And what ends up happening is they will kind of cluster up on themselves and precipitate out. It's kind of one of those you can't uncook the egg situations. Using heat is actually one of the very effective ways of disrupting those interactions and denaturing a protein. So cooking an egg is a really good example of protein denaturant. So when we have an egg, the albumin in the egg white, is a protein rich environment. And when we heat it, the proteins begin to unravel. And that is why when you cook an egg, you go from like runny to like a solid, um, not not clear, um, egg, is because we've denatured the proteins and they're precipitating out. Other ways we can denature proteins: ultraviolet light sometimes, strong acids and bases. We talked about the idea that amino acids and their Zwitter ions kind of act as their own acid and base. However, all acids and bases, or excuse me, all buffers have thresholds that they can't go over. We can also have heavy metal poisoning, so lead poisoning, things like that, are also uh, possible sources of denaturant. Okay, this ends chapter 19. Please go through and review the amino acids. Make sure you're comfortable with those. Make sure you're comfortable with writing the backbone for an amino acid, as well as knowing primary, secondary, and tertiary structures of proteins.